Hello everyone, this video is part of YouTube lecture series initiated by Zoological Society of Assam. Myself Ashish Sarkar presenting life cycle and pathogenicity of Taenia solium. Taenia solium or pork tapeworm is an endoparasite of man. The life cycle of Taenia solium is indirect. It passes through pigs as intermediate host and then to humans as definitive host. Disease due to the tapeworm Taenia solium occurs in two forms, Taeniasis and Cysticercosis. Let's move to the historical background of Taenia solium. At the end of the video, we will have clear knowledge on history, geographical distribution, habitat, and reproductive system of Taenia solium. The life cycle and pathogenicity of Taenia solium will also be cleared. Human Taeniasis as well as porcine and human cysticercosis caused by the pork tapeworm Taenia solium are ancient diseases. The Ebers papyrus includes descriptions of tapeworms. Both eggs and cysticerca of Taenia solium have been found in the intestine and stomach of Egyptian mummies. Intestinal tapeworms were a common knowledge among Arabian and Egyptian physicians, who treated them with pumpkin seeds, an herbal medicine that is still used nowadays. Confirmation that Taeniasis and Cysticercosis were caused by the same parasite was provided during the 19th century by German pathologists. Soon thereafter, the knowledge on the life cycle of Taenia solium was completed by experiments in Belgium and Germany. These findings were further confirmed by the seminal work of Yoshino, who infected himself with Taenia solium cysticerci to study the life cycle of the cystode. Let us discuss the geographical distribution of Taenia solium. Taenia solium is cosmopolitan in distribution. It is found in all those parts of the globe where pig is domesticated. It is particularly prevalent in developing countries where pigs are raised in poor sanitary conditions. It is common in areas where animal husbandry practices are such that pigs and cattle come into contact with human feces. In the Western Hemisphere, it is mostly found in South and Central America. The tapeworm is also widely found throughout countries in Africa and Asia. However, the number of infections is low in populations of Muslim and Jewish cultures where eating pork is forbidden. Adult tapeworms are found firmly anchored in small intestine of human host. Its nutrition is saprozoic and respiration is anaerobic. Proglottids are full of eggs and these proglottids are removed from the body through feces. Onchosphere is found in pig intermediate host. The habitat is gut and tissue of pig host. Taenia solium is hermaphrodite. Each proglottid develops a complete set of male and female sex organs. This gives the worm an enormous power of multiplication, which is very essential for a parasitic animal. As the worm is protandrous, in the first mature proglottids, only the male sex organs are functional, and in the rest, female organs also become functional. In the remaining proglottids, the sex organs progressively degenerate, except the uterus, which is correspondingly enlarging to almost fill the proglottids. Male reproductive system comprises of numerous small rounded testes scattered almost throughout the parenchyma. A fine canal starts from each testis. This is called the vas efferens. The vas efferentia from the adjacent testis unite to form larger canals, which ultimately open into a thick convoluted duct, the vas deferens. The vas deferens starts from the middle of the proglottid, runs outwards and open by the male genital pore into a cup-shaped chamber, the genital atrium. The terminal part of the vas deferens is modified into a straight muscular protrusible copulatory organ, the cirrus. The female reproductive system comprises of a large ovary that lies in the posterior half of the proglottid. The ovary consists of two rounded or oval lobes joined by a narrow bridge or isthmus. Each ovarian lobe is composed of a number of small radiating follicles. Nearly from the middle of the isthmus there extends backwards a short oviduct. It joins with spermatic duct and opens into the ootype. Ootype is the chamber where fertilization takes place. There may be self-fertilization or cross-fertilization between the segments. Vagina is a tube that connects genital pore to the ootype through which the sperm enters. At the inner end, it contains seminal receptacle and spermatic duct. Uterus is a wide, straight, simple, blind tube arises from the ootype and extends forward almost the whole length of the proglotid. Its beginning is very narrow and is often termed the uterine duct. The eggs are stored here after fertilization in the gravid females. The ootype is surrounded by unicellular Mealy's glands that open into it and receives the vitelline duct from a large lobulated vitelline gland situated behind it. They release their secretions through their ducts into the ootype. In the left-hand side image, we can clearly see a whole tapeworm. The tapeworm's body is divided into four parts: the anterior part, immature proglotids, mature proglotids, and gravid proglotids. The anterior part has been shown in detail in the right-hand image. We can clearly see rostrum, hooks, and suckers by which the tapeworm gets anchorage on the intestinal wall of the host. In that image only, a complete proglotid has been shown along with its internal reproductive system. The difference between mature proglotids and gravid proglotids is that mature proglotids have well-developed sex organs, but gravid proglotids have lost their sex organs, but their body is full of eggs. Taenia solium life cycle includes man as definitive host who harbors adult worms and pig as intermediate host which harbors larval stages. Pigs get infection by ingesting eggs with contaminated water and food. Man acquires infection by eating undercooked pork containing encysted larval stage. A man harboring the adults may auto-infect oneself. The whole life cycle is studied under definitive host and intermediate host. Humans, the definitive host of Taenia solium, are colonized by the larval stage, the Cysticercus. When a person eats undercooked pork containing these Cysticerci, they actually eat measly meat. Measly meat is the meat that contains Cysticerci. The Cysticerci are enclosed in protective sacs. Thus, the stomach's acid digests the meat, but the larval forms of tapeworm make it into the person's intestinal tract unscathed. When pork containing Cysticercus is consumed with inadequate cooked pork, the larvae are digested out of the meat in the stomach and duodenum. Once inside the small intestine, the inverted scolex of the Cysticercus everts out. This process of evagination is stimulated by bile juice and digestive enzymes. Using the scolex, it anchors to the intestinal wall. It grows in size using nutrients from the surroundings. Its strobila lengthens as new proglottids are formed at the foot of the neck. In 10 to 12 weeks after initial colonization, it is an adult worm now. After growing into adult, they perform copulation and fertilization. Fertilization is internal in Taenia. Therefore, it is always preceded by copulation. There is usually a single tapeworm in one host so that only self-copulation is possible. It can occur in two ways. Cirrus may be passed in the vagina of the same proglottids and spermatozoa shed there. The strobila is folded to bring the distant mature proglottids opposite to each other, and cirrus of an anterior proglottid penetrates and inseminates the vagina of a more posterior proglottids where ovaries have also become functional. Fertilization is self in Taenia, but when eggs and sperms come from different proglottids, it is often considered as cross-fertilization. Cross-fertilization is perhaps more common as the testes and ovaries usually do not mature at the same time in the same proglottid. The spermatozoa swim through the vagina by lashing their tail and reach the seminal receptacle. Fertilization occurs in the ootype. Now we have capsule formation and their laying. The fertilized eggs acquire yolk cells from the vitelline gland. From the vitelline duct, a thin, delicate shell forms round the zygote along with yolk cells. The structure thus formed is termed as capsule. When complete, the capsules pass into the uterus. Secretion of Mealy's gland acts as a lubricant to facilitate the passage of capsules into the uterus. As more and more capsules reach the uterus, it enlarges and develops 7 to 13 pairs of lateral branches for their storage. The gravid proglottids detach from hind end of strobila by apolysis, either individually or in groups of 2 or 5, and are passed in feces of the host during defecation a few times in a week. Freshly passed proglottids perform wriggling movements by muscular contractions and squeeze the capsules so that they are well scattered. Development of zygote begins in the uterus of the worm, and by the time the proglottid is shed, it has already changed into an embryo. The zygote by total and unequal cleavage produces a mass of blastomeres. This mass includes numerous micromeres, few mesomeres, and 2 to 3 macromeres. The micromeres form embryo with 3 pairs of claw-like hooks at its posterior end. This 6-hooked embryo is known as the hexacanth. The mesomeres and macromeres form the inner and outer embryonic membrane respectively round the hexacanth. The inner membrane later hardens into a thick, chitinous, radially striated secondary shell or embryophore. The hexacanth with embryophore, outer embryonic membrane and capsule wall is termed the onchosphere. The onchosphere loses the capsule wall and outer embryonic membrane and is left with the embryophore. It does not hatch from embryophore until eaten by a suitable intermediate host, mainly pig. It remains viable for several months on the soil. In this image, we have a young onchosphere and a matured onchosphere. In the young onchosphere, we can see the outermost cover, that is eggshell, beneath which thick chitinous radially striated embryophore is visible. Vitelline cell is also observed. Morula is a stage of embryo formation. In the matured onchosphere, all the structures are same except the morula has converted into hexacanth, which is characterized by the presence of 6 hooks. The second phase is intermediate host, which involves post-onchospheral stage of Cysticercus. Being the first step, infection of intermediate host is important. The post-onchospheral development of the larva proceeds within the intermediate host, the pig. Pig, by virtue of its coprophagous feeding habit, acquires the infection by ingesting the host stool along with the onchospheres present in the dislocated gravid proglottids or lying freely in the feces. After ingestion, onchospheres hatches out and penetrates the gut wall. It is carried in the systemic circulation. The naked onchospheres are filtered out from the circulating blood into the muscular tissue. During this stage, the parasite does not attain sexual maturity. Rather, they are transformed into Cysticercus stage. Next transformation is from onchosphere to Cysticercus. The onchosphere quickly changes from a solid larva into a bladder form filled with fluid and having a group of cells that will differentiate further into an invaginated scolex. When the freed onchosphere enters the intestinal wall, it is very minute. At about 6 days, the metacystode is still solid. It has an outer membranous wall comprising of pleomorphic cells, while its inner contents are myxomatous. By 12 days, the metacystode is larger and becomes 60. Between 20 and 30 days, a rudimentary scolex is visible. Hooks appear by 40 days and the rostrum and suckers are distinguishable by 40 to 50 days. The metacystode reaches its fully grown size by 60 to 70 days. Now let us discuss the structure of Cysticercus larva. The Cysticercus is an ovoid bladder stage. A mature cyst is milky white bladder surrounded by a fibrous capsule and contains an invaginated scolex. It is also known as bladder worm. It contains a thick fluid rich in protein and salt. The bladder consists of outer and inner layers. The outer layer has characteristic hair-like processes. This layer not only plays a protective role but also serves as a trophoblast that absorbs nutrients and excretes metabolites. The survival time of a cysticercus is limited to a few years. The naturally degenerating cysticercus becomes necrotic and eventually gets calcified. or forms a granuloma that finally transforms into a fibrotic scar. In this image, we can clearly see a free hexacanth which is found in the gut of a pig when the pig eats a onchosphere. From that onchosphere, this free hexacanth is developed. The hexacanth then undergoes invagination and attains a bladder-like structure. In the latter images, we can see the development of proscolex from the bladder. Cysticerci actually occur in three distinct morphological types, of which the most common is the ordinary Zoology Cysticercus, with an invaginated scolex and a fluid-filled bladder. The intermediate form, with a scolex, and the racemose, with no evident scolex, are much larger and more dangerous. They can measure up to 20 cm and contain 60 ml of fluid. Up to 13% of patients may have all three types of cysticercus in the brain. As we all know that humans get infection when they eat pork having cysticerci, known as measly meat. But let us discuss now what will happen when human ingest eggs of Taenia solium. In this case, humans may be also a secondary host when they ingest food and water contaminated with eggs due to person's unhygienic habit. Onchosphere penetrate intestine and develop cysticercus in man's eye, brain, etc. It is the dead end of the tapeworm's life cycle and dangerous for the man too. However, usual secondary host, that is pig, stores the cysticercus in muscles which when ingested by man develops adults in man's gut and continue the life cycle. In this image, a complete picture of life cycle of Taenia solium has been shown. In the intestine of human being, we can clearly see an adult tapeworm. The adult tapeworm liberates the proglottids having full of eggs. When the humans excrete these eggs into the environment, pigs feed on these feces and gets infected. Inside pig's body, the hexacanth develops into cysticercus which stores in the muscle. When a person eats such kind of meat known as measly meat, the person develops inside its stomach again the pork tapeworm, that is Taenia solium, and in this way the life cycle continues. But if accidentally other person eats the eggs from the environment of Taenia solium, the person will develop cysticercosis in his body's vital organs like brain, eye, muscles, etc. Now let us discuss the pathogenicity of Taenia solium. Taeniasis. The adult Taenia solium in man causes the disease known as Taeniasis. The adult worm, in spite of its large size, causes surprisingly little inconvenience to the patient. When the infection is symptomatic, vague abdominal discomfort, indigestion, nausea, diarrhea alternating with constipation and weight loss may be present. Reduction and occlusion of the lumen of intestine also takes place, so the free movement of foodstuff is disturbed. Occasional cases or acute intestinal obstruction, acute appendicitis and pancreatitis have also been reported. The hooks and suckers of tapeworm may cause mechanical irritation. Infection with larval stage is known as cysticercosis. When humans ingest food and water contaminated with eggs, cysticerci evade host defense. But when a cysticercus dies, it explicits more inflammatory response. Cysticercosis occur in any tissue. Subcutaneous tissue and muscles are common. Eyes, brain, liver, lungs may also be invaded. Due to the presence of larvae, cellular reactions start with infiltration of eosinophils and other WBCs, which calcify the larva, leading to the death of the larva. Subcutaneous nodules are asymptomatic. Muscular cysticercosis may cause acute myositis. The more dangerous neurocysticercosis cause epilepsy along with intracranial tension, psychiatric disturbance, etc. Ocular cysticercosis are found in vitreous humor, conjunctiva, and subretinal space of eye. This may cause loss of vision or blurred vision. Here are the references of the presentation and suggested readings for the student community. Maximum information in the presentation has been taken from the book Non-Chordates, Protist to Pseudocoelomates by Manideep Raj. Thank you very much for being here till the last of the video. If you have any feedback, then please contact me. My email ID has been given here. Thank you very much once again.